right? I hope I'm audible to all. So today we are talking about applications of electrophoresis. So we have talked about electrophoresis, that is gel electrophoresis, then agarose gel, as well as the polyacrylamide gel also. So when we talk about electrophoresis, the very first purpose when it comes to applications is what? To separate the charged particles, charged molecules, charged species, right? So when you are applying electric field, under applied electric field, charged species will move and uh, they will get separated, and you will be able to characterize these molecules on the basis of mass and charge, right? So that is very basic principle that when we talk about electrophoresis. So when it is to applications of electrophoresis, very first is separation of biological molecules which are charged entities, and of course it is very important that relatively lower molecular masses such as amino acids, as well as those which are having high molecular mass like proteins, polypeptides, DNA, RNA, can be very easily separated when we are talking about electrophoretic methods. So we are just trying to summarize all the applications. So besides separation, it is there as an analytical tool also. When I say analysis, the very first thing is about what is the relative molecular mass of a protein or a DNA that comes to your mind, right? So when we talk about the electrophoresis as a technique to find out the molecular mass of any sample, say it is a protein, for example, right? So if I want to find out molecular mass of a protein which I am working with, how should I proceed, and how it can be considered as an application of electrophoresis? So of course we know that SDS stage is the electrophoretic process which is going to separate individual polypeptide chain, and of course the movement of the protein is due to the charge which is developed on the protein on account of SDS. Protein has its own charge, but when you treat it with SDS, again being the detergent ion, it is going to charge the entire surface of protein with sulfate ions which are going to get exposed. So minus 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 charge, and protein is linearized form that we know. We have done SDS in details. Now the thing is, the surface charge of these protein complexes per unit area is constant. So regardless of the charge of the individual polypeptide chain, individual polypeptide chain will have its positive or negative charge. True, but when you treat it with SDS, it will be uniform, and it will be uh, per unit area. It will be constant. These are the two things. Now you can talk about the protein complexes to assume their shape as a long rod shape, which is denatured protein now, and entire mobility of protein now is not because of the charge, but because of the molecular size of the polypeptide, right? The factors that the base of molecular weight determination by SDS gel electrophoresis is that the entire protein is now with negative charge, and per unit area of the protein, the, the charge, surface charge is constant. So if the area of the protein changes, the total net charge will change. So ultimately what is the factor? It is molecular weight, molecular mass of the protein that is going to decide its mobility. So can we use this as a method to determine molecular weight of a new protein, right? So of course it is in the gel, so sieving effect is going to be there, and a linear relationship exists between the electrophoretic mobility and the SDS protein complex. Right. So when you are going to talk about the SDS protein complex molecular weight and electrophoretic mobility, yes, they are related. So you can see in this plot here relative migration distance. That is one thing. And on the other side, what you have is log molecular weight, right? So as the log molecular weight increases, the mobility is going to be less, so the distance traveled will be less. And if the molecular size is log molecular weight is less, the distance traveled will be high. That is how are we going to get the relationship between molecular weight and the that of the mobility. So when you talk about mobility, just imagine this is first one, second protein, third protein, fourth protein, fifth, like that you have 7 8 proteins here. And just imagine the protein, say, whose molecular weight is about 6 into 10 raised to 4, right? Or here in this case it is 2 into 10 raised to 4. So when I say 2 into 10 raised to 4, and this size, it is comparable to say zero point 0.7 into 10 raised to 4. That is how if I go. Now, highest the protein mass, highest the molecular size, highest the molecular weight, it is going to migrate slowly, and the mobilities will be less, right? So this graph demonstrates how you can explore the linearity of mobility of the protein which is treated with SDS and its molecular weight. So if you have these mixture in which you have series of proteins which are also SDS treated and whose molecular mass is known to you, right? And one among those is unknown one which you are going to run. So when you do the electrophoresis, all are going to get separated depending upon their electrophorate mobilities, and you can find the distance traveled. All right, and the distance traveled is plotted against log molecular weight. Here this horizontal line, which is, or you can say the perpendicular line on the graph line, is telling you my sample protein is having somewhere 1.3 around, say log molecular weight is 1.3 into 10 raised to something. So this, if I extrapolate it onto the graph, I can find out the uh, distance traveled, and vice versa. Because I don't know molecular weight, I want to find out molecular weight, so what I can measure is only the distance traveled on the gel. So that is put on x-axis. So this is the vertical graph, vertical point on the graph, which I am correlating with the graph point, and it is correlated with the y-axis. So on y-axis, I have log molecular weight. So I can find out based on the distance traveled, the point is trapped on the graph, and it is extrapolated on y-axis. So what I see is, okay, the protein sample is having the size about 1.4. So this is how, of course, these are imaginary numbers. You can have a standard sample mixture, say like myoglobin, glutamic dehydrogenase, aldosase, trypsin. You can take these. So if I say glutamic dehydrogenase, for example, so you are going to have its molecular size around 5 into 10 raised to 4. Aldosase, it is around 4.2 into 10 raised to 4. Or you say trypsin, it is around 2.4 into 10 raised to 4. Myoglobin also is about 1.8 into 10 raised to 4. So known molecular sizes are there. I'll get a graph line or trend line which is telling me based on their mobility and molecular weight, this is going to be the pattern. And when I run my sample molecule, what I get only the distance traveled on the gel I can measure. So from that distance traveled, I can plot a perpendicular line on the graph and find out what is its relative molecular weight. So electrophoretic systems, they are used for finding out the molecular weight of the sample analyte. Alright, you agree this? Alright, now let's go to um, another application. Now here I'm talking about sequencing gels. What are sequencing gels? We have talked about um, a restriction digestion, we have talked about SDS page, we have talked about special gels which are used for sequencing. We have learned in previous years, that is NGS technology, right? Or Maxim Gilbert method or Sanger's method, right? So at the end of that, what do? You find out the sequence, right? Now, but sequencing gels are those, I mean, the extended fragments, for example, Sanger sequencing you are doing. So different sized fragments which are terminated at DDNTP addition, they are generated, and you are going to separate out those segments by electrophoresis. So ultimately, when you talk about determination of DNA sequence, it comes electrophoresis. So we have learned maybe Maxim Gilbert method or Sanger's method of dideoxynucleotide. What we do is we are going to take help of high resolution polyacrylamide gel electrophoresis at the end, separate out the strands and find out the sequence. Now, both these methods, may it be Sanger's method or may it be Maxim Gilbert method, both depend on generating specific set of radiolabeled fragments, and each is going to terminate at a particular base. So when you use high resolution polyacrylamide gel, then the fragments differing by a single nucleotide can be resolved. So what is the key point? Fragments which differ by a single nucleotide base can be resolved with the help of sequencing gels. Right. Now, next point is about when it comes to applications, northern blotting and southern blotting. We know what blotting does mean, right? So when electrophoresis is done, bands are separated. You are going to transfer these components in the band to the filters or as cellulose acetate paper, nitrocellulose paper, different types of membrane filters are there. Onto that you are going to transfer it, and then you are going to say ki you have blotted it, and then you are going to analyze with labeled probes. That is how we generally do. So when we talk about application of electrophoresis, we can talk about northern and southern blotting. Here I have taken a slide directly from Thermo Fisher, as you can see the label, right? Purification of DNA and RNA, digestion of the DNA is done. Then you are going to do the gel electrophoresis, and then the separated fragments of DNA or RNA, they are transferred onto the nylon membrane. All right. Then what you are going to do? Pre-hybridization. What does it mean? You have already done it in TY. You have done it in last year also. It is kind of a revision and compilation of the points. So I'm going little hurriedly. So you are going to wash the nylon membrane with pre-hybridization solution containing uh, the, uh, the DNA, so that it is blocked non-specific DNA interactions, and you so that your response is clear. There is no noise, right? It is required. So preparation of a probe is required. You are going to add the probe, which will selectively bind, and then you can see if it is giving you the fluorescence or if it is giving you the radioactivity. Depending on the probe used, you can detect the signal, you can detect the response. And if it is giving the response, that means hybridization has happened, and the particular molecule of your interest, the DNA sequence of your interest, is present. Or when it is western blotting, particular protein is present. So RNA, DNA and protein blotting you have learned. So in blotting you are going to separate it, whether it is DNA, RNA or protein. You are going to separate it by gel electrophoresis. Then you are going to transfer that material by blotting onto nylon membrane. Then you are going to incubate it with radioactive probe, for example. Then you are going to allow the binding of probe to the specific DNA RNA sequences, and then it is taken for analysis. So depending upon whether it is autoradiography, whether it is fluorescence technique, so visualization will be accordingly done. So when you talk about application of electrophoresis, you can very well write southern, northern and western blotting as a method for as a method where electrophoresis is of importance. So when you are checking the complementarity of two DNA strands, yeah, right, or you want to find out whether the DNA or the mutation in the DNA which are which you are interested, are they present in it or not, that also can be detected by these techniques. So sequence of a portion of the desired DNA is known, then you can synthesize the complementary oligonucleotide, radiolabel it, and make it react to the sample. And the sample is been separated by agarogel electrophoresis, transferred to nitrocellulose paper, and then you can take it for autoradiography if it is radiolabeled. That is how we can just summarize southern and western blotting technique, which is considered as application of electrophoresis. Right. Now here is another point, that is restriction mapping. The word itself tell you there are two things, restriction and another is mapping. When I say restriction, of course restriction enzymes are there, and mapping is finding the position, right? So when you are talking about say the study related to genomic or clone DNA, it is required to have some sort of map, right? To know the positions, to differentiate one area from the other. So one technique, that is restriction mapping, and use of gel electrophoresis, again without which you cannot progress, is of importance. So how do we do it? Digesting the DNA with different restriction endonucleases, then those fragments are going to be separated by maybe agarose gel or maybe polyacrylamide gel electrophoresis. Now you know when to choose agarose and when to choose polyacrylamide gel. This we have discussed in previous lectures to very much of details, right? Depending on size and porosity of the gel and size of the fragment, size of the molecule, you have to decide, right? So separation is done by electrophoresis. Then visualizing these fragments while they are in the gel with the help of ETBR, right? We are talking about DNA, and then estimating their size in relation to the standard run on the gel simultaneously, right? That is what is the ladder that you say, isn't it? So you can take help of restriction enzymes, then generate the fragments, then these fragments are run over agarogel electrophoresis, they will give you bands. So these bands are compared with the ladder bands, the band generated by the ladder, and then you can find out the relative size. You can estimate the relative size of the molecule. So that is what is restriction mapping of the DNA. That is one of the applications, right? So how do we progress? Say for example, it is 100 kb DNA. You are going to treat it with say three different restriction polymerases. I'm sorry, restriction enzymes, and here it is, for example, SAL1, then it is BAM-H1, like that, right? And here is a mixture of two. So DNA that was 100 kb is fragmented into different size fragments depending upon the site of action of these enzymes. And after that, you have to run polyacrylamide gel, right? So this is only treated with SL1. This is with BAM-H1, and this is the mixture of the two. So treatment with one enzyme will give you, say, for example, three fragments of three different size. Treatment with balm H1 is giving you two fragments. One is very big, one is very small. That's why they have run at different rates. So one is just very close to the initial point and another has run to the end of the gel. And here is the third one, which is having mixture of the both the enzymes, right? So you are getting another pattern. Here is the marker or the ladder where the size of the fragments is known, and you have the ladder generated. Now by comparing, can you find out the relative molecular sizes of the fragments? Usually you can, right? So you can construct the restriction map as well as you can find out the or estimate the size in relation to standard. So if as an example of application of electrophoresis, if you just write key restriction mapping of DNA and draw the schematic diagram, it will be more than enough. There is no point in describing the techniques in details because you will not be able to complete your paper in that case, right? Each technique we have discussed earlier, but when it comes to writing as a summary of applications of electrophoresis, you can take help of such schematic diagrams, right? So now restriction mapping, when I say the advanced version of restriction mapping is called optical mapping, and it is used for larger genomes. So fragments, bacterial genomes or small size 100 kb fragment we had taken. So small fragment, it is digested to even smaller fragments and you have done restriction mapping. But higher version, advanced version of the technique, when you are going to handle larger size genomes, is referred to as optical mapping. In this, DNA fragments are not separated in agarose after digestion. Restriction digestion is done by placing the isolated chromosome in agarose solution on a microscopic slide and allowing a gel to form. When the agarose forms the gel, DNA is stretched, right? And the restriction enzyme is flooded on the agarose gel containing the DNA and the mixture is incubated. Then the restricted DNA is observed under high power microscope and the relative location of the restriction sites are visualized as gaps. From the location of gap, the restriction sites are mapped. So it is advanced version optical mapping. So when you say restriction mapping of DNA, you are handling a specific size of uh, genomic sequence. But when it is entire genome analysis, then the advanced version is referred to as optical mapping. Right, now here further we go about applications, right? So very commonly known diagnostic tools when you talk about in biochemistry, electrophoresis is one of those, right? So here is the example which is showing the case of thalassemia, right? So hereditary abnormalities are there, maybe in the hemoglobin, for example, and there are two types when hemoglobin variants are there. So abnormal hemoglobin are there, and thalassemia is there, right? So hemoglobin electrophoresis is done to identify the abnormality. So HB electrophoresis is very easy, sensitive, and fast method at pH 8.5 using agar gel with cellulose assisted membrane in routine analysis. It is done. Now, the protocol, of course, is electrophoretic process, right? But it is talking about hemoglobin, so it's a protein. Fine. Now, what we are talking, it as an application. We are not asking you the method. We are to just refer to the method for its purpose of application. So what you have is the bands which are generated, that are those are compared here in this diagram. What are those bands? You have 1, 2, 3, 4, 5 bands, right? So these horizontal bands are nothing but the gel strips, right? So this gel 1, gel 2, gel 3, gel 4, gel 5. Now, what are these gels for? The first gel, or you can say first well in the entire gel. See, blue is considered as a single gel, and these are the different wells. That is how you can look at it. That is not very important. What is important is to see this is the sample from normal person. This is thalassemia. This is beta thalassemia major, right? This is sickle cell test, and this is sickle cell trait and sickle cell anemic trait. So the hemoglobin, when it is run on electrophoresis, the bands what you are going to get. So this is a starting point, origin point, HbA2, HbF, and HbA. These are the molecules which are monitored. So if you see as against that of normal, right? So you have origin point, you have HbA2, you have HbF, and you have HbA. What is the case in beta thalassemia? This is a very broad band, right? Are you able to see this? Fine. Similarly, you are having a additional band in sickle cell conditions, right? So some of the cells which are sickle celled, the hemoglobin content, you know the variation, you know the genetics behind it. So the protein is going to have been different from the normal protein, normal hemoglobin, and what happens is you are going to have a separate new band that is appearing in place of in middle of the gel, and this HbA1 is missing in case of sickle cell anemia trait, right? So this is very self-explanatory picture actually when you talk about comparing the profiling of hemoglobin by electrophoresis method. What kind of band formation is observed? Are the bands matching with the normal, or are they deviating from the normal? If there is deviation, what is the position of this band, and is it matching with the trait, right? This is the trait. So if there is HBS present, a band will be observed here. So this is the standard reference, and against that you are seeing the sample from the patient, and it is showing you a very broad band here. That means if you correlate these two, it indicates presence of HBS, right? So in diagnostics also you can take help of electrophoretic technique, right? Now when you are talking about various applications of electrophoresis, here is another one. A complexation of two or more molecules you can find out with electrophoresis. What is that? That is, for example, if you have nucleic acid drugs and separating the mixture on gel electrophoresis that you want to do. So, if a protein is there and a molecule which is targeting that protein is there, it will interact with it. Similarly, you can take example of DNA, where DNA is the target and the molecule that is drug probably is targeting that DNA. So when these DNA and drug molecule interact, or protein and drug molecule interact, the properties of a molecule as a whole are altered because it's a mixture of the two now, right? So the complexation what you are going to get Can it be revealed? Can it be analyzed? Yes, it can be analyzed for by electrophoretic technique. So here is an example which talks about analyzing interaction between corona proteins and nanoparticles. So electrophoretic mobility shift assay is the name of it. Okay, shift assay matlab the position of the protein is going to get altered when it reacts with the ligand, when it react with the target drug, or when it reacts with any metal ion, so on and so forth. So a band that a protein would form, and a band which is the protein, the band which is the protein has formed is at one location, but when the protein gets complex with another molecule, like say drug molecule, then this complex is going to get positioned at different place. So you can visualize what kind of effects are there and what is the complexation product is. And for that you are going to use uh, EMSA, that is electrophoretic mobility shift assay. That means electrophoretic mobility of the protein will get altered when it is complexing with some other molecule. So here is an example. So here are the nanoparticles which are going to carry the drug molecule, right? And these are the protein solutions. So when they are mixed, this is the target protein against which this drug molecule is designed. So these are going to be put together and they are incubated, right? And analysis of protein shift that you are going to do. That means you are going to do electrophoresis, and in one well you are going to put protein alone, and in another one you are going to put the protein which is complexed with the drug molecule present in the nanoparticle, right? Electrophoresis is allowed to go, right? Native page electrophoresis you are seeing here. Then you are going to go for staining is done with either Comasi blue, or you can go with silver staining or fluorescent staining. We have learnt all these staining methods. So after staining, what you see is there are two different bands, the native protein alone and the complex protein, the protein that has interacted with the drug molecule. You have to check whether the target that you have chosen and the drug molecule that you want to apply against that, are they interacting or not, are they complexing or not, what kind of complex is getting formed. All this analysis needs to be done. So electrophoresis would help in finding out the complexing reactions, and that is how done with EMSA, as that is electrophoretic mobility shift assay. What is the shift? This is the band form when it is only protein, and this is the complex when the band formed is due to the complex between nanoparticle carrying the drug and the protein analyte, right? So because the complex is now heavier than the protein alone, so it has not migrated to the distance compared to the protein alone. Not necessarily always it will be so apart, so far away. They may be next to each other also, but they will not be exactly matching. So you can find out the complexing reactions, interactions between two different molecules which are mixtured together, right? So this is another application of electrophoresis process. Of course, when you talk about mm electrophoresis for nucleic acid, their separation on the basis of size, finding out their structural conformational details, that is single stranded DNA, dsDNA, and circular DNA. The mobilities are going to be different. Again, with the help of ladder, you can find out or estimate the size of the fragment, right? You can detect the presence of the particular protein recombinant molecule if you have synthesized, whether it is getting expressed. So you have extracted the protein and put it on electrophoresis, and you are looking for the presence of protein in the host plant or host animal, whatever it may be, or host cell culture also it could be. So overall, it is a kind of analytical tool, right? So to summarize, I would say you can use electrophoretic processes when it comes to applications in molecular biology alone. You can say determination of DNA sequences, southern northern blotting, right? Then restriction mapping of DNA, then DNA footprinting, and also you can say variations in conformation of nucleic acids. They can be analyzed using electrophoresis. When it comes to a study of application to proteins, the very first one that we talked was about the determining relative molecular mass, right? So you can find out the uh, uh, molecular weight of proteins by gel electrophoresis, where you are using a where you are using a reference proteins, finding out their mobilities and mobility of your unknown protein, and comparing it on a graph. On the graph, you have x-axis mobility on electrophoretic gel, and on y-axis you have molecular weight. Of course, it is log molecular weight value, because the range of protein molecular masses are going to be too diverse. So you have to use the log molecular weight as a indicator, right? So you can find out molecular weight of many proteins. I mean, has been determined using this method, right? It is very simple method. Reproducible results are there. It is very easy to construct and interpret the results, right? So when it comes to electrophoresis, this was the first lecture to summarize some of the applications of electrophoresis technique.